Hello, my name is Brendan Scott. I was nominated to talk about cyanobacteria, even though I struggled to pronounce some of the names, because most of my cultivation experience is with cyanobacteria. I studied chemical and biological engineering at Colorado State University, and then I worked trying to convert algae into biofuels. I had the chance to work with several companies focused on this goal, including Solix, Algenol, and Synthetic Genomics. I also worked with the U.S. Department of Energy in monitoring their algae and waste-to-energy portfolios. In this role, I was exposed to many different algae cultivation teams and paradigms. Through collaboration with the Algae Technology Education Consortium, I've been given a chance to help prepare this online class, and I've learned even more about algae and its uses in that process. I hope you learn a lot too and enjoy this just as much as I have. Thank you, and now we'll get started. This is a biochemist's rendition of the sugar glucose. In this shorthand, every bend in a line is a carbon atom. Carbons are numbered from the first carbon on the oxygen in the ring in a clockwise manner. In glucose, there are 6 carbon atoms. This is another version of glucose. Both versions have 6 carbon, 6 oxygen, and 12 hydrogen atoms. What is the difference? In glucose on the left, the oxygen is attached to the carbon number 1, is below the plane of the ring. In the other glucose, the oxygen attached to carbon number 1 is above the plane of the ring. The one with the oxygen below the ring is alpha glucose. The one with the oxygen above the ring is beta glucose. An easy mnemonic reminder is ABBA, like the 70s Swedish supergroup. Alpha below, beta above. This nomenclature is only used to describe the position of the oxygen's carbon number 1. This is ribose, a 5-carbon sugar. Is it alpha ribose or beta ribose? The oxygen on carbon number 1 is above, so it is beta ribose. Carbohydrates are polymers of simple sugars like glucose and ribose. Placing another alpha glucose next to the one already there orients the oxygen on the carbon number 1 of the molecule on the left next to the oxygen on the carbon number 4 of the glucose on the right. A condensation reaction, removing a water molecule, links the 2 glucose molecules together via what is called an alpha-1,4 linkage. This is one of the most common linkages in carbohydrates. Another alpha glucose above the first one will orient the oxygen of carbon number 1 of the sugar above the oxygen of carbon number 6 of the one below. A condensation reaction creates what's called an alpha-1,6 linkage. Alpha-1,6 linkages are less common, but are necessary to put branches into the carbohydrate molecules. This cartoon shows the 2 major carbohydrates in starch. Amylose, which is a long straight chain of glucose monomers linked by alpha-1,4 linkages, and amylopectin, a much larger branched carbohydrate that has alpha-1,6 linkages every once in a while, making it branched molecule of chains of glucose with alpha-1,4 linkages. These molecules are very stable and can store energy for long periods of time. As we just saw, 2 alpha glucose molecules next to each other can be linked by an alpha-1,4 linkage. If we add a third glucose, they are also linked by an alpha-1,4 linkage, and so on. Placing 2 beta-glucose molecules next to each other presents a problem. The oxygen on carbon number 1 of the glucose on the left side is above the plane. The oxygen on carbon number 4 on the right is below the plane. How are the oxygens placed close enough for the condensation reaction to occur? Flip the glucose on the right upside down. Now the condensation reaction can happen, linking the 2 sugars with a beta-1,4 linkage. Adding another beta-glucose orients the oxygen so the condensation can happen. The next glucose must be flipped over again, and so on. The upper glucose polymer is called amylose, a major portion of the starch in your French fries. The lower glucose polymer is called cellulose, the major molecule in wood. Are you surprised to learn that baseball bats are mostly sugar? Compounds with very different chemical properties can be made from the same monosaccharide. It just depends on how they're put together. There are two major functions for carbohydrates in algae. The first is as an energy storage molecule, or reserve carbohydrate. Algae are classified by the different types of reserve carbohydrates and the location in the cell where they are stored. The cyanobacteria store their energy in starch that has alpha-1,4 linkages with alpha-1,6 branch points. The starch is stored in the cytoplasm. As shown in the legend in the lower left corner, alpha-linked carbohydrates are represented by green ovals and beta-linked carbohydrates by yellow ovals. The blue and purple hexagons represent two different structures that house carbon dioxide concentrating mechanisms. These structures increase the efficiency of fixing atmospheric carbon into sugars. Pyrenoids are subcellular compartments found in the chloroplasts of many algae that are associated with concentrating CO2 to fix carbon from the air into sugars. Carboxysomes are protein complexes found in the cyanobacteria and glycophytes that have the same function of concentrating CO2 to fix carbon. These are not the same structures, but they have the same function. The chlorophyte green algae store energy in starch with alpha-1,4 links and alpha-1,6 branch points. In green algae, the carbohydrates are stored in the stroma inside the chloroplast. This makes sense because according to the endosymbiotic theory, chloroplasts are descended from free-living cyanobacteria. The endosymbiotic theory states that eukaryotic organelles, such as the chloroplast, evolved from bacteria trapped inside the eukaryote. The trapped bacteria provided a useful function for the eukaryote, in this case producing sugar from CO2 and sunlight, and these symbiotic bacteria have since lost the ability to live independently of the eukaryote. We will devote a lecture to the endosymbiotic theory and the origin of chloroplasts later. The rhodophyta red algae also have chloroplasts derived from cyanobacteria, but they use a slightly different form of starch called Floridian starch that also is linked by alpha-1,4 linkages and has alpha-1,6 branch points that are about every 10 glucose monomers apart instead of around 20 glucose monomers like starch in green algae. Their starch is stored in the cytoplasm, not in the chloroplast. The glaucophytes are a third eukaryotic algae that have a chloroplast derived from cyanobacteria. They store their energy in alpha-linked Floridian starch like the red algae, and also store the carbohydrate in the cytoplasm like red algae, but they have a carboxysome in the chloroplast similar to the carboxysome in the cyanobacteria. These three phyla are examples of primary endosymbiosis. The eukaryote organelle derived from a symbiotic relationship with a bacteria. Secondary endosymbiosis occurred when another eukaryote took in a eukaryote that had an organelle derived from primary endosymbiosis and kept the organelle. Brown algae obtain their chloroplast by endosymbiosis of a red algae. The carbohydrate used for energy storage is called laminarin and is a beta-1,3 linked carbohydrate and branches at beta-1,6 linkages. Laminarin is stored in a membrane enclosed vacuole in the cytoplasm. The euglenids arose from a secondary endosymbiosis of a green algae. They have a carbohydrate called paramylon that is linked by beta-1,3 linkages and is stored in the cytoplasm. The chloroarachnophytes also have chloroplasts derived from the chloroplasts in green algae. The carbohydrate is a beta-1,3 linked molecule that may be related to paramylon. This carbohydrate is stored in a membrane enclosed vacuole in the cytoplasm. The red algae have been used by many other algae types as the origin of their chloroplast. The haptophytes have a storage sugar similar to the laminarin in brown algae called chrysolaminarin. Chrysolaminarin is also a beta-1,3 linked carbohydrate and has beta-1,6 branching. It is also stored in the membrane enclosed vacuoles in the cytoplasm. Diatoms use the same carbohydrate and storage location as the haptophytes. There are other significant differences between the brown algae. Haptophytes and diatoms that we will cover in later lectures. Chrysophytes also use the same carbohydrates, except it is not clear if the carbohydrates are stored in a membrane-bound vacuole in the cytoplasm or are free in the cytoplasm. The dinoflagellates store their energy in alpha-1,4 linked starch in the cytoplasm. Cryptophytes are similar to the dinoflagellates in the type of starch, but their chloroplast is enclosed in an envelope that is vacuole-like. The starch is stored in the space between the chloroplast and the outer envelope called the periplastid space. They also have a pyrenoid in the chloroplast. Here's a summary of the starches, types of linkage between sugars, and the carbohydrate storage locations in the various algae phyla. Since many use the same starch and have the same carbohydrate storage location, why are they classified differently? Reserved sugars are not the only diagnostic feature used in classifying algae. We previously looked at pigments, and now we'll look at structural sugars.